It was good. Today we got diversity of life. So yeah. So what are the Linnaean taxonomic divisions? So what was the mnemonic? King, Philip. So we have kingdom. Yo, ain't no way I already forgot. King Philip. Dear King Philip came over for good soup. Dear domain. Okay, domain, kingdom. Fennel? No. Phylum. Okay, wait. Dear King Philip came over for good soup. Dear King Philip came over for good soup. Dear domain, kingdom. Fennel. What is the word for it? Faunal? Phylum. Domain, kingdom. Yo, I already forgot. Faunal? Phylum. Dear King Philip, domain, kingdom, phylum. Dear King Philip, class, order, family, genus, species. Who let him cook, bro? I only took one time. What are the two structures used for adhesion in prokaryotes? It says pili and fimbriae. What is present in the prokaryote cell wall or bacterial cell wall? That's peptidoglycan. How do prokaryotes reproduce? They use binary fission. Binary fission? It's just creating identical copies. What is the flagella made up in prokaryotes? It's made of the flagellin protein. 2 ways to classify life and how they obtain energy. So first we got chemoautotrophs and then we got photoautotrophs. Chemoautotrophs, they produce their own energy through chemical substance, while prokaryotes, they obtain it from light energy. What is a parasite? A saprocyte. So a parasite is a kind of 2 ways. We have autotrophs and heterotrophs. That's my bad. And then autotrophs, you can be chemoautotroph or photoautotroph, which is the next question. So a parasite is a type of heteroautotroph, lives in the host and takes energy or what car is that, bro? It takes like food and like energy and nutrients from the tissue of the host. Saprophyte is a decomposer and it just takes nutrients and energy from dead material. Difference between photoautotrophs and chemoautotrophs, like I said, photoautotrophs use light energy, chemoautotrophs use chemical energy. Classify the relation of oxygen. So we have obligate aerobes, which is that they require oxygen. Then we have obligate anaerobes, which they cannot be in oxygen. And then we got facultative, which is they can use both oxygen and non-oxygen. Compare domain archaea to others. What separates it from domain bacteria? So I'm not sure about 6 of them, but one is that they have introns, so that's similar to eukaryotes. What else? They have their own membrane lipids, I believe, rather than ester. I think they have ether or something. Unique rRNA, extremophiles, no peptidoglycan. Damn. Yeah, I wouldn't have known that. What are the 3 types of archaea? We got methanogens, which is they produce methane, like CH4. Then we got, well, it's just on my mind, bro. Like thermophiles, which is like they like heat. So we got 2 thermophile. Then we got So what did I even say just now? Thermophiles, methophils? Wait, hold on. No, not methophils. Three types of archaea. Interesting. How do Methanogens, thermophiles, halophiles. Oh, they like halogens. I see. Halophiles, thermophiles, methanophiles or something. Yo, this exam is cooked, bro. Methanogens. Okay, so it's not phile because they don't like it. It's that they produce it. I should have known that. So peptidoglycan cell classification. So we got positive and negative. Gram positive, gram negative. So gram positive, that's pink, right? Right, 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 right. Okay, so gram positive, yeah, gram positive is purple, gram negative is pink. So gram positive is pink. Wait, no, gram positive is purple, gram negative is pink. So it has a thin peptidoglycan. Wait, bro, wait, I need to think about this. Gram positive is freaking purple. What's the color of the stain? That's how I remember it. So gram negative is pink. So if you're negative, that means you don't get the stain, kinda. So that means you have thin peptidoglycan layer, and then you have lipopolysaccharides which produce endotoxins on the outside, and then you do not have telcoic acids. Whoa, hold on a second. And then you stain purple, right? No, you stain pink. Wait, hold on. Okay, so let's go under the influence that the stain is purple. So if you're gram negative, you're gonna turn purple. You're gonna turn purple. No, you're gonna stay pink because nothing changed for you because you're negative. What am I even trying to say, bro? Okay, let's talk about this again. Let me just stop itching my eye. Okay, so we have two classifications of bacterial cell wall. You can have gram positive or gram negative. Gram positive means that you're pink. Gram negative means that you're no. Gram negative means that you're pink. Gram positive means that you're purple. So let's say the stain is purple, then you're gonna get infected by it and you're gonna become purple. But if you're not, then you're gonna be purple. So you're gonna have a thick cell. You're gonna have a thin peptidoglycan layer if you're gram negative. So you're gonna be thin peptidoglycan. So you have thin peptidoglycan layer. You have an outer lipopolysaccharide with endotoxins on the outside. You're gonna stain pink, and you're gonna have no telcoic acid layer. Then if you're gram positive, that means you're gonna be purple. Gram positive purple, you have a thick peptidoglycan layer with no outer lipopolysaccharide layer, and you're gonna have a telcoic acid layer, which is between the peptidoglycan and plasma membrane. That's what stains purple. The telcoic acid is what stains purple. Okay, that was rough. Oh, I already talked about it here. We can just delete this one. Okay, what are endospores? Those are just dormant bacteria cells that activate when the conditions are right. They can withstand crazy conditions. Nomenclature of bacterial shapes. So we got the cocculi, then we got the spirali, and we got the, what's the last one? Spiral, cocculi, and baculi. Oh my god, look at me count. Oh. So it's not cocculi, it's actually called coqui. Coqui. Okay, so we got coqui, bacchi, baccali, coqui, and spirally. So coqui is spear shaped, baccali is rod shaped, spirally is spiral shaped. Bacala, okay. Then we have groups. We have grouping. You could group them into two. You could do them linear chains or you can do them in clusters. What is the name for clusters? Yo, this is like not registering. Strepto, and then staphylum. Okay, so you can have three shapes: cocky, spirally, and bacterially. Backly. Okay, backly, spirally, cocky, so bacterially, backly is rod shaped, cocky is spear shaped, spirally is spiral shaped. But I already forgot the names of the groups, bro. So you can have clumps or linear. So you can have strepto or staphylum, strep throat, strepto or staphylum. Okay, let me cook up again. Nomenclature of bacterial shapes. So exhibit A. So you can have backly, which is rod shaped. You can have cocky, which is spear shaped, and then you can have spirally, which is spiral shaped. Then you could have. Yo, I already forgot about it. Strepty. All I remember is strep throat. Okay, strepto, staphil, strepto. Okay. So there's classifications, there's nomenclature. So number one, you can have backly, that's rod shaped. You could have cocky, which is spear shaped, and you can have spirally, which is spiral shaped. Then you could have strepto, which is strep throat, which is linear. Then you could have staphil, which is clusters. Yes sir. Oh yeah. So two common types of bacteria. There's nitrogen fixing bacteria, which is a mutualistic relationship with plants, and you can have cyanobacteria. Cyanobacteria are also called blue-green algae. However, they're not algae because that would imply that they're eukaryotes. They're bacteria, so that's something to know. So what are the three types of genetic recombination? So you could, I'm trying to block out the, let me just cover it. So there's conjugation, transformation, and transduction. So what are the three, what does that mean? Conjugation is with the pilus, transformation, conjugation. I'm like 99% sure conjugation with the pilus. So it's like you like stick it into the other cell and then you put the plasmid in. Then you got transduction, and then you got transformation. So one is when you heat shock it and then it just absorbs the protein. I believe that's transformation. Then transduction must be, let me think, bro. How else would you get the DNA into the cell? Oh, you could use the virus. So I want to say that's transduction. So the transfer of conjugation is considered to be, I don't know. So where is it talking about this now? Damn, wait, why are these notes so much information that I never heard about? What are we talking about? Fungi. Oh, this is 11.8. I haven't even learned about this. Gonorrhea. Thermophiles, halophiles, obligate, facultative. Yo, I'm locked in, bro. I need to learn this, bro. Let me just look on these. Conjugation, DNA transfer, transformation, DNA uptake. So transformation is DNA uptake. Transduction is when the virus. Okay. So I think I cooked that up. Okay, flashback. What is one I struggle with? So the prokaryotes use cilia and fimbria. Cilia and villi? No, fimbria and pill? No. I want to say cilia and fimbria. Oh, well, I was wrong. It's pili and fimbria. So I was right with the pili, I guess. So that was not good. What are the Linnaean taxonomic divisions? So King. Wait, what's his name? Dear King. What's his name again? Philip. Dear King Philip. So domain, kingdom. Yo, how did I not remember that? Fennel. I keep on thinking about freaking organic chem, bro. The trauma. Fennel. Faunal. Fizzle. No, bro. Okay, dear. Let me just get that. Dear King Philip. Class, order, family, genus, species. But what's the P for? What is P? Phylum. Phylum. Okay, compare domain archaea to others. What separates them from domain bacteria? So they have introns. They have no peptide glycan. They have their own kind of lipid membrane bonds. I think they have ethers rather than ester. Yeah, so three types of archaea, they're methanogens, they're freaking thermophiles, and they're halophiles. Nomenclature of bacteria shapes, so you got cocci, spirali, bacilli, then we have, bro, lock in, bro, strepto, sacrophil, bro, no. Sacrophil, no. Bro, what's the name of it? Strepto, okay, strepto, one of them. Strepto is linear, then what's cluster? Oh my god. Staphil, but it's genuine rage bait, bro. What the frick? Okay, anyways, continuing on. So, what transfer is conjugation considered to be? Transfer of DNA, I guess. Horizontal transfer, I remember that. Okay, how does transformation occur? So transduction involves virus, right? Yep, and then transformation occurs with heat shocking or making the cell compatible so that it'll just take in the information. So yeah, okay, that's all. Okay, we're making progress, I guess. The rest is about viruses. Are viruses included in the taxonomy grouping? No, they're not. They're non-living. What genetic material are viruses made of? They can be made of RNA or DNA. Viruses have a cover called a capsid, which is made of a capsomer. So it's a protein capsid, so the polymer is made of capsomer. What does the virus take from the host? So when it breaks free out of the host, it'll take some of the outer membrane with it. It also, I guess, you could say, hijacks the machinery in the cell, and it'll make its own stuff. But it also does take out some of the outer cell wall, like the plasma membrane. How do lytic viruses work? So they're gonna go inside the cell and they're gonna replicate, and then they're gonna lyse out of it. They're gonna explode the cell. How do retroviruses work? So they're gonna do reverse transcription. So they're gonna go into the cell and then they're gonna hijack the machinery and they're gonna make DNA out of RNA, and then lysogenic viruses, they'll go in, then they'll integrate themselves as part of the DNA. Then the cell will continue to divide, and then the virus will still be in all those cells, and then it's one day it's gonna be like, psych, you thought he was gone, and then it'll be like, hell nah, and then it's gonna start spreading everywhere and stuff like that, and it's gonna kill all the cells, and like, got you there. All right, so yeah, what are the basic structure of the virus? So we have the hereditary information, so like DNA, RNA, and then we got the caspid, which is like the outer coating, which kind of protects it. So yeah, nucleic acids and protein caspid. So what do bacteriophages have? So they have legs and they have something else. They have okay, they have tall tail fibers and a sheath. Okay. Damn. What are virions? Virions? 3 ways to release the viral particles. So you could do, I want to say like, okay, so there's apoptosis, which is when the cell just explodes. Then you can have exocytosis. Yeah. So exocytosis will just leave. Then you can have budding, and you can have apoptosis. What are virions? I'm not too sure. Complete virus particles. Yeah, that's I'm not sure. Okay, 2 types of viruses. So you can have there. Okay, so there's one called a eukaryotic thing, and the so it's prophage and provirus, I think. So prophage is for prokaryotes. Provirus is for eukaryotes. 3 ways bacteria defend from viruses. So one is like Casper-Combs or something. It's called Kriper-Cas system. That's where the prokaryotes will like take in the cell, like the virus, and they're gonna incorporate it into their DNA kind of, and then they're gonna like recognize it, and then every time it comes in, they're gonna be like, yeah, you thought, and then it's gonna like destroy it because it's gonna recognize it. It'll keep like a copy and be like a vaccine kind of. There's also mutated cell wall, like cell membrane, so it'll like recognize that it's a virus and it'll be like, nah, you're not coming in this time. So yeah, and then the other one is, what I say, CRISPR-Combs, the friggin' cell wall, and then restriction enzymes. I'm not sure how that works. Restriction enzymes. Well, restriction enzymes, they break down. So I think it would just break down the RNA or the genetic material of the virus until it's non-readable. 4 types of viral infections in humans. So there's onset, like there's acute, which is like you get sick and then it becomes worse, and then you reach the peak and then it goes away. There's chronic, which is when it'll like have an initial like really bad phase, and then it'll just like, you know, chill out, but you're gonna still have that kind of acute kind of thing. There's latent, which is when it'll like it'll come and go. It'll like be like, yo, I'm so sick, and then it'll be like, oh, I'm gone now, and then it'll be, oh, I'm so sick, and then like, oh, I'm so. And then slow, I would say it's like, it's kind of like how the other type of, what's that type of virus, lysogenic, it'll just chill in there, and then it'll be like one day be like, psych, I've been here the entire time, and then it'll just start like infecting you. 3 types of viral outbreak. Well, there's pandemics, epidemic. So pandemic is like a worldwide thing. Epidemic is within a population. And then there's endemic, which is like a very small, I would say, endemic. I don't really remember that one. Okay, honestly, I don't fully remember these ones, so let's come back to them at the very end to like kind of review them. How do vaccines work? So vaccines will introduce like a kind of dead version or like an inactive version of the virus so that your body can recognize it. It'll activate the immune system, and then your body will be able to recognize it and know how to treat it. Why do we get infected sometimes? So mutations occur with these viruses. You know, they can combine, which I'll get into later, but they can kind of like have different antigens, and they'll start being different and special. 2 types of changes. So there's like transgenic, I think. I don't remember this one either. 2 other types of infection particles. Okay, so there's prions and then viroids. Okay, I was trying not to look at it. So prions are like cooked proteins that will make other proteins get cooked. So it's kind of like mad cow's disease. And then viroids are just modified RNA that like kind of mess things up. So pyroids they have 2 characterizations. One is that they're really hard to get rid of. Like they like under heat, they won't like get messed up. Then the other one, I'm not too sure. resistant to enzymes. I'm not too sure about that. What are the two types of changes to which species are infected? Bro, who be driving, bro? Antigenic shift and antigenic drift. What the frick? Okay, so antigenic drift is it'll infect the same host species, so it's like mixing. Antigenic shift is like two will combine together and it'll increase the host ranges. Okay, interesting. Prions and viroids. So what are the two classifications? Long incubation, so they'll act slowly, and resistance to deactivation. Okay. So let's continue on. So phylum is the word. Okay. Then there's strepto. Bro, there's no way. This keeps on slipping my mind, bro. Strepto, strapto, and streptostaphylum. Okay, streptostaphylum. Three types of archaea, methanogen, halophiles, and thermophiles. What separates it from domain bacteria? It's just like, okay, wait, let me talk about gram-positive and gram-negative again. So gram-positive and gram-negative. So positive means you become purple. Purple means you have the telcanoic acid. Right. Positive means you stain purple. That means you have a thick peptidoglycan layer. No, that means you have the telcanoic acid, which is between the peptidoglycan layer and the plasma membrane. So you have a thick peptidoglycan layer. So then the opposite, which is gram-negative, is pink. It'll have a thin peptidoglycan layer, have lipopolysaccharides on the outside, which release endotoxins, and then you're gonna have no telcanoic acid. Okay. Nomenclature, yeah, bro. Frick that, bro. What are virions? Yo, I don't understand what virions are actually. Let me control F that. It's not findable. It's not present. Perhaps it's not even a thing. 3 ways to release the viral particles: budding, exocytosis, and apoptosis. What is the difference between prophage and provirus? Provirus is for eukaryotes, prophage is for prokaryotes. 3 ways to defend from viruses for bacteria. You got the CRISPR-Com, which is when like it's like kind of vaccines for bacteria. Then you got mutated like or evolved cell membrane. So it's like it'll like deny it from entry, and then what was the other one? Restriction enzyme. I'm trying to find the notes for 11.6. I can see you wiggle, push. Protists. I didn't learn about 11. Well, this don't talk about freaking viruses, bro. Perhaps it was not mentioned in this notes. That's quite annoying, bro. Intro to viruses is page 21. But why is it the last thing? Why can't I scroll down? Viruses. Yo, this is not in order, bro. It's kind of annoying me. Wait, what? Oh yes, here we are. Retrovirus, bacteriovirus. Oh, so retroviruses go from ssDNA to dsDNA. Oh, so there's lytic cycle, lysogenic cycle, which I already talked about, epidemic, endemic, pandemic, cell surface mutations. Okay, so it prevents phage from binding to it. CRISPR-Cas thing, that's just the other thing. Bacterial enzymes that can recognize and cleave specific viral. Okay, so it's just breaking it down. Antigenic shift. Okay. 4 types of viral infection in humans. We already talked about that one, right? Yeah, I'm not even trying to find it. Lysis. Damn, so these placenta? What? Why are we talking about the freaking body? What the hell? Okay. Okay. So we have genetic drift and genetic shift. I mean, antigenic shift and antigenic drift. Accumulation of small mutations resulting in minor changes. So antigenic shift is what it'll infect more. Drift is like, okay, shift is like shift, like I'm shifting my population. Drift is like drifted into like a small mutation or something. You know what I'm saying? Okay, so prions. Prions are misfolded proteins that cause other proteins to get misfolded. One is that they're really hard to like denature. Like they're like freaking they're able to stay at this high temperatures, and then the other one is that they last a long time. Like, you know, like they're just like there for a long time. Viroids are small RNA, ssRNA that infect plants, and then they'll cause diseases. So yeah, that's pretty much all for today. See you, goodbye.